All right, today we are going to be talking about the molecular structure of DNA and RNA. And specifically in this lecture, we are going to be looking at both the structure of a nucleotide. So in terms of learning outcomes, you want to be able to describe the structure of a nucleotide, and I also want you to be able to compare and contrast the structures of nucleotides found in DNA versus that of RNA. So, the main repeating structural unit of both DNA and RNA is the nucleotide. And we've discussed DNA and RNA before. We've referred to it as a polymer, and those individual subunits, again, are called nucleotides. Nucleotide has three main components. The first component is a phosphate group. That is followed by a pentose sugar, pentose meaning it has 5 carbons. This is where DNA and RNA are different. So in RNA, we have ribose as the pentose sugar, and in DNA, we have deoxyribose. We also have a nitrogenous base, which is where we get the A's, T's, G's, and C's. So let's take a closer look. So here's the phosphate group. We have a phosphate in the center surrounded by oxygens. There is a double bond to one of those oxygens. The sugars, we have 5 carbon sugars, so 1, 2, 3, 4, and 5. Down here, 1, 2, 3, 4, and 5. Here we have all of the different bases. Again, both DNA and RNA have the A's, G's, and C's, or adenine, guanine, and cysteine, or cytosine. And DNA has thymine, and RNA, in place of thymine, has uracil. So let's take a closer look at how these pieces fit together here. So the phosphate group is attached onto carbon number 5. When we form a chain, the other side of this phosphate is attached to carbon 3 of the preceding sugar chain. And this will be a little bit clearer when we actually look at an actual chain. So just know here that DNA has a directionality. There is one end of a chain that has a 5' end, one end that has a 3' end. And in between the sugars, you have a phosphate group. So it'd be phosphate group sugar, phosphate group sugar, phosphate group sugar. Now, we also have the nitrogenous bases here. And those bases are fixed onto the sugars at position 1. All right, so here we have position 1 on deoxyribose, and here's position 1 on ribose. And that connection is to where I have these blue arrows highlighted on the nitrogenous bases. So here, the sugar is attached to this nitrogen, and here on adenine, it's attached to this nitrogen. Here it is attached to this nitrogen, over here to this nitrogen, over here to this nitrogen. So all of these are covalently bonded onto these ribose sugars at position 1 and linked to a nitrogen. Now, I would like you to be able to recognize these different bases. Okay? You don't have to be able to draw them from scratch, simply be able to recognize them. And so what I'm going to do is kind of walk you through how to tell the difference. Now, you should be able to deduce here that there are some that are bigger, have a 2-ring structure, and some that are smaller that have a single ring structure. Okay? The double ring structures we call purines, okay? And the single ring structures are called pyrimidines. easy way of trying to remember which one is which. Here we have a short word for a big structure, and here we have a big word for a smaller structure. Now, if you can remember that purines are adenine or guanine, the easiest way of telling them apart is to take a look at what's happening up here at position 6. So in guanine, we have a carbonyl group, or double bonded oxygen. And on adenine, we have an amino group. Now, if you take a look at the pyrimidines, one of the first things to look for there is what is attached to carbon number 4. Okay, so in cytosine, it is an amino group, and in both thymine and uracil, it is a carbonyl group. Okay. Now, if you can remember that you have DNA versus RNA differences between thymidine and uracil, provided that you know you're looking at DNA or RNA, you will be able to tell which one is which. Okay. There are other differences as well that you can kind of key in on. So coming back here to purines, if you look at positions 1 and 2, you can see some differences there. So taking a look at position 2 here, we just have a hydrogen. Here at position 2, we have an amino group. And consequently, over here, we have a nitrogen. The nitrogen is a part of the ring, so when we have these ends embedded into the corners of these shapes, it means that that's a nitrogen, not a carbon. And you can see this nitrogen has a hydrogen bound to it. This one does not have one because there's a double bond here. Over here on the pyrimidines, we can also see differences in position 3. Again, both thymidine and uracil, we have a hydrogen at position 3, and for cytosine, we have that same scenario, a missing hydrogen because there is a double bond over here that replaces that bond. All right, finally, if you are having trouble telling the difference between DNA and RNA and can't figure out if it's a T or a U, you can always take a look at position 5. Position 5 has a methyl group for thymidine, and it just has a hydrogen for uracil. Now, another easy way of telling DNA and RNA is taking a look at carbon 2 of the sugar ring. So if you look here at ribose, you can see you have an OH group, and this fits with what we know about carbohydrates. For carbohydrates, every carbon is linked to an oxygen, usually in a hydroxyl, but sometimes in a carbonyl. So here we have 1 in an hydroxyl, 2 hydroxyl, 3 hydroxyl, 4 we actually have that carbonyl, so this is forming the ring structure, and then 5 hydroxyl. Notice that in deoxyribose, we are missing that oxygen. So it's almost like we deoxygenated it. Now you get the idea of what the name is, right? So take a look at position 2, see if there's a hydroxyl there. Ultimately, this plays into the stability of DNA versus RNA. We'll talk more about that later. All right, so again, I want you to be able to recognize, but you don't necessarily have to be able to draw them. All right, putting them all together, here again we have the phosphate group. It's attached to carbon number 5. We have the base attached to carbon number 1, and for deoxyribose, so deoxy, so it's missing the oxygen at position 2. And for bases, we can have A, G, C, or T. For RNA, we have that same structure here. We have that phosphate group attached to carbon number 5. And over here at position 2, we have the oxygen. And in terms of bases, we have A, G, C, or U. All right, now, we refer to these individual subunits as nucleotides, but sometimes that phosphate group is missing, and we have a separate name for when it is just the base and the sugar. When it is just a base and a sugar, we refer to it as a nucleoside with an S. When we have a phosphate group attached as well, it is called a nucleotide with a T. Now, we can have different numbers of phosphate groups attached, linked in a chain onto here. So we could have another one out here and another one out here. So if we have just a single phosphate, we'd refer to it as, in this case, if we have adenosine, we would have adenosine monophosphate, meaning one phosphate group, or diphosphate for when we have two, or triphosphate. And here you might recognize this as our familiar friend ATP, which is one of the universal energy sources. It is also the same thing as a component of DNA. So again, just to kind of recap this, we have the nitrogenous base attached to sugar 1, 1, 2, 3, 4, 5. On 5, we have the phosphate groups. If it is missing the phosphate group, so just the sugar and the base, it is a nucleoside. If we have a phosphate group on it, then we refer to it as a nucleotide. If it's just one, it's monophosphate, 2, diphosphate, and 3, triphosphate.